students, this is SriRam Shastri. I once again welcome you back to my new video session. So this video is continuation of the male reproductive system from the last video session. So as you can see on the board, we are going to discuss today about the diagrammatic representation of male reproductive system, and one by one we'll discuss the parts of it. This is the lateral section of testis. So starting with the main diagrammatic representation of male reproductive system. So it's also called as urinogenital system. The reason why is it called as urinogenital system is male reproductive system is connected to the excretory system. So here you can see this is the urinary bladder. This urinary bladder, below the urinary bladder itself you have one pair of glands called seminal vesicles or seminal glands. These seminal glands are connected to the vas deferens. Vas deferens is connected to the epididymis. Epididymis is connected to the testis. So internal structure includes seminal vesicles, one pair of seminal vesicles, then one circular gland called the prostate gland. This one is called as prostate gland, and one small pair of Copper's gland are also called as bulbourethral gland. Okay, so this cylindrical structure is the penis, and this bulged end is called as glans penis. It is covered with a skin called as foreskin or prepuce. Okay, this tract is the urinary tract itself. This is nothing but urethra. Okay, and urethra, it forms a narrow region surrounding the prostate gland, forms the ejaculatory duct. Okay, so these are the scrotal sacks. In the scrotal sacks, testes are being situated. So the testis, the structure of testis we'll discuss here. The diagram is just beside it. We'll discuss it later. So this diagrammatic representation of the male reproductive system is like this. One by one, all the parts we have to discuss in detail. So first we'll start with discussion of the structure of testis in males. Okay, so as you can see on the board here, this is the lateral section of the testis. Okay, here it will be very small to draw, so I have drawn it very separately in a large portion so that you can observe it very carefully. So this layer of the testis, externally it is covered with the two layers of connective tissue called as tunica vaginalis. So tunica vaginalis is the outer connective tissue layer covering the testis. The inner fibrous connective tissue covering the testis is tunica albuginea. This is tunica albuginea. Then the vascular tissue which is connected to the testis is tunica vasculosa. So outer layer is tunica vaginalis, outermost layer. Middle layer, which is fibrous connective tissue, is tunica albuginea, and inner vascular tissue is tunica vasculosa. So these three layers are the coverings of the testis. Inside the testis there are around 200 to 300 compartments. These small pink lobules, what you can see, compartments, there are around 200 to 300 testicular lobules. They are commonly we call them as compartments. They are called as testicular lobules. Okay, so each testicular lobule consists of around 1 to 3 seminiferous tubules. Seminiferous tubules, these yellow structures, what you see here, these are seminiferous tubules. Seminiferous tubules are internally lined by the germinal epithelium, which differentiate into spermatogenic cells and undergo spermatogenesis to produce sperms. Along with germinal epithelium, the seminiferous tubules also contain Sertoli cells. So we'll discuss that in another diagram with the testis. That is, this is lateral section. We'll take the transverse section of the testis to study the structure of seminiferous tubule later. So for now, just we'll understand the major structures what are present inside the testis. Then if you talk about the compartments, I said there are 200 to 300 testicular lobules. Each lobule consists of around 1 to 3 seminiferous tubules. The length of seminiferous tubules is approximately 70 to 75 centimeter. 70 to 75 centimeters in length. These all seminiferous tubules, they collect at one region. This region, what is you see here, a network of seminiferous tubule, is called as rete testis. From the rete testis, a single tubular structure arises. This single tubular structure which arises is called as vasa efferentia. Vasa efferentia is connected to again a very long coiled structure inside the structure called epididymis. That vasa efferentia is super coiled inside the epididymis. Okay, so this region outside the testis, what you see, is called as epididymis. Epididymis can be divided into three portions. Upper head part are called as caput epididymis. this one center part, body of epididymis or corpus epididymis, and the lower tail part, or you can call it as caudal epididymis. Caudal epididymis gives rise to a single tube called as vas deferens. Okay, so sperms are produced inside the seminiferous tubules. They are collected and temporarily stored in the epididymis. So epididymis is divided into three parts. This vasa efferentia is supercoiled inside the epididymis. So approximate length of it is around 6 meters. Approximate length of it is 6 meters. And after that there is a small tube which arises from the caudal epididymis called as the vas deferens. This vas deferens is connected here in this diagram. You can see this is the vas deferens. It is connected to the seminal glands, right? Seminal vesicles. From there, so mixture of sperms and seminal vesicles, they are being removed out as semen, which is called as ejaculation. The process of release of semen is called as ejaculation. If it occurs inside the female body, it's called as insemination. Ejaculation and insemination. Release of semen is ejaculation. Release of semen into the vaginal canal is called as insemination. Clear? So this is what is the structure of lateral section of testis. This is lateral section. In another video, we'll take up the transverse section of the testis to study the structure of seminiferous tubules. For today, we'll stop this here. Thank you.